So the first part of this figure is going to be the part that was on the top. That's figure 5A. As I said before, the figure A shows the immunoblot, and this is going to be with the anti-LIMK1 and the anti-LIMK2. And that can be seen up on the figure A. We've got the control on the left, and then the siRNA LIMK1 and the LIMK2. And so basically the HeLa cells were transferred with an siRNA pool to interfere with the expression of the LIMK1 and 2. And so the protein cell extracts were analyzed with this immunoblot that you can see on here. Based off of the control that you can see in this figure, it can be seen that there is a dark band, which indicates that the protein is present, whereas the absence of a dark band would mean that the protein is not present. The last part of this figure is going to be figure B. So this is showing the confocal images of the immunostaining, as you can see above here. The HeLa and the protein cells were co-transfected with the GFP cells together with the siRNA against the ILMK1 and the ILMK2. They were then treated with doxycycline, noted as DOX, DOX on the figure. Those confocal images of this immunostaining are shown with the GFP siRNA is in green. The F-actin is in red, and the nuclear host staining is in the blue. Okay, so the first part of this figure 6 is going to be part A. So this figure shows the distribution of the cells in the multiple cell phases. So we've got G1 and G2 are the gap phase. S is the synthesis phase, where DNA replication occurs, and M, mitosis, where the cell divides. Those three cell phases of the cell cycle are shown after the DNA staining. The HeLa cells were transfected, again, with the encoding P57, the protein, with the CDK inhibitor. It can be observed from this, you can see in the figure that the control and the one with the CDK inhibitor, they have similar percentages of the DNA content, both at roughly 73%, and the protein has the highest percentage at 83%. So the next part of this figure is going to be part B. This figure shows an immunoblot with the anti-protein, the anti-MYC, and the anti-phosphocophilin antibodies. And then down at the bottom, you can see the G3PDH, and that was used as a standard for equal loading of the protein. In this process, the HeLa cells were transfected with the expression vector that encodes that protein, the alternative to the protein with the CDK inhibitor, both alone and in combination with the MYC and LIMK1. It can be seen on this immunoblot in the figure B that the phosphocophilin, the two at the bottom, and this G3PDH, once again, the two at the bottom of the immunoblot, they have dark bands. The phosphocophilin definitely has more of a defined band than all the other ones, and that can kind of be seen in this figure.
So now that Cher and Morgan have done a good job just talking about all the figures and going through the results, I'm just going to summarize what they said and talk a little bit about what was covered in the conclusion. So in figure 1, it talked about how P57 increases the actin stress fiber formation. And then going from that, figure 2 talked about how P57, the enhancement of that expression increased the LIMK1 expression, which was the phosphorylation enzyme, or the phosphorylation kinase. Figure 3 talked about how P57 was regulated to the nucleus and the cytoplasm whereas LIMC1 was just in the cytoplasm. This is just how the researchers were trying to figure out where P57 was in regards to the cell and where LIMC1 was in regards to the cell. Figure 4, it talked about the phosphorylation activity of LIMC1 and that when it was increased, that the phosphorylation activity of LIMC1 was increased when P57 was co-expressed. And it also talked about how the phosphorylation activity was due to P57 and it was not related to ROCK phosphorylation, and that it was also not related to the threonine at position 508 phosphorylation. Figure 5 talked about LIMC2 and how it did not affect P57 activity or the actin cytoskeleton, which showed that P57 activity was just in regards to LIMC1 and not LIMC2. Figure 6 talked about losing the CDK inhibiting ability. That losing CDK inhibition of P57, when it lost that ability to inhibit CDK, it did not affect the increased activity of LIMC1, which was just talking about how the P57 activity in regards to LIMC1 was not related to P57's ability to inhibit the CDKs. Figure 7 talked about how cells expressing P57 recruit more actin towards stress fiber formations, which reduce its ability to recruit actin to other parts of the cell. Figure 8 it was just demonstrated that cells expressing P57 exhibit decreased cell migration motility abilities. And this was in regards to the wound closing experiment. So in conclusion, the experiment confirmed that P57 is a multifunctional protein that is involved in cancer suppression or growth, and that patient outcomes in regards to P57, it would depend on what type of cancer and where it is. P57 was also thought to maybe be involved in the intrinsic apoptotic pathway, and this was found that when apoptotic stimuli were put into the cell, P57 was regulated to the mitochondria where it is involved in the intrinsic apoptic pathway. This experiment demonstrated that in cancer cells, specifically the HeLa cells, when P57 is regulated to the cytoplasm and co-expressed with LIMK1, it causes decreased motility and invasiveness in cancer cells. And they also mentioned that future studies need to be conducted to identify additional roles of P57 in tumor suppression.